incredibly grateful to Lucy and the Science Festival organisers for giving me that small boy at Christmas feeling that I still get whenever I'm asked to stand up and talk about one of my favourite subjects. But I have to tell you, ladies and gentlemen, that I'd only spent 2 or 3 minutes thinking about what it was I'd signed up to this evening before I began to get seriously cold feet about the whole thing. And to explain or to illustrate why that might be, I need a little bit of audience participation. And one of the whole ideas, of course, about Science Week is to get the audience involved. So I need all the help I can get from you guys and girls tonight. But first of all, could I have a show of hands, please? Anybody in the audience who would not object to being called a biologist of some description, please raise your hands. This isn't a trick question. I'm not going to point to you gleefully and say, "Ha, you can give the talk." Actually, maybe that's not a bad idea. But hands up, anybody who's a sort of vaguely card-carrying biochemist, biologist, bio, anything you like. Fine. So I reckon out of, I don't know what the audience is, but maybe 150 odd, we've probably got something like about 10? Something like that. So you see immediately the problem, almost the biggest challenge as a lecturer you can face. You've got a wide disparity of knowledge, if you like, of what you're going to talk about in the audience. So that's really very tricky. I could actually ask, not just biologists, but anybody like to volunteer a comment on what you're looking at in the movies that are on the screen in front of you? Anybody at all, biologists or non-biologists? Yes, Joe?
Cells?
Yeah, you're looking at cells. Can you do any better than that?
Cells, molecules and cancer?
Unless I'm very much mistaken, that's a young gentleman from St Faith's who's going to go far. Could a biologist enlarge? One word?
Cell division.
Say it again.
Division.
It's in part cell division.
Tubules.
You're really looking at tubules, and in particular you're looking at a protein sort of continuously being broken down and reassembled in the cells. And we'll come back to that protein in a minute. It's actually a thing called tubulin. So to get back to our challenge, the question really is how to keep everyone happy, and maybe the title of this slide should have a question mark after it. But what I'm going to do is to spend a few minutes right at the beginning by talking about cells making new cells, making copies of themselves. A phenomenon that I will try to refer to as proliferation throughout this talk. However, it is in biology very frequently also called multiplication, and it's quite often called division, and so it's the only branch of science where multiplication equals division, and you can see from that why we're the despair of our colleagues in the hard sciences. But in talking about this question of cells making copies of themselves, is really going to the heart of cancer. Because if you wanted a one-line, actually almost a 3-word description of cancer, it would be cells proliferating abnormally. Cells making more and more of themselves, either in the right place, but at the wrong time, or at the right place and the right time, but maybe just doing it a little bit more quickly, a little bit more efficiently than normal, or of course making more of themselves somewhere where they shouldn't. And so cell division, multiplication, proliferation really lies at the heart of cancer. But it's actually almost impossible to talk about that for more than a minute or so without mentioning, of course, DNA and protein. So I'm going to spend a couple of minutes after that on what's sometimes called the central dogma of molecular biology. DNA makes RNA makes protein. And in doing that, I'm going to say a little bit about what proteins in particular are, and really how it is that they are so astonishingly wonderful and really are the reason for all of the life forms on our planet. But as soon as you start talking about proteins, in the context of cancer, it is but a small step to think about what can go wrong with them. And that, of course, is mutations. Mutations in the first instance in DNA, but mutations in this context because it is mutations that are the driving force behind cancer. So all the abnormal proliferations that are part of cancer are fueled by mutations in our DNA, very often, though not always, reflected by mutations in proteins. And then what I'm going to do is to turn to what I like to call the greatest revolution. And by that I mean the greatest revolution, certainly in biological science. And that's the sequencing of the human genome. And I'm going to talk about that a little bit to try and illustrate why it is that it has already, in just 10 years, completely transformed the way we think about cancer and indeed the way we go about treating it now. And one corollary of that is the evolution of new cancer drugs, and I'm going to talk about one example of that rather briefly. But I'm then going to make the point that this is not entirely a garden full of roses, so to speak. In other words, that there are some very serious problems that confront us at the moment in terms of dealing with cancer. And I'm going right at the end to talk about, if you like, a rather radical way of thinking about cancer therapy and show you some results, admittedly thus far only in mice, that perhaps offer a new way forward. And indeed, some of those results have not even been published. I want to start at the beginning though, and I'm going to show you now a very short, about 20 seconds or so, animated movie, and I need a bit of audience help from that because I want somebody to explain what we're looking at. So here we go. I suspect pretty well everybody will know what's going on. Okay, what have we just seen? Anybody? We've seen fertilization and we've seen cell division. So we've gone from one cell being fertilized by an egg, one cell, one egg being fertilized by a sperm, to this thing that looks a little bit, it's generally held like a mulberry, so it's called a morula for that reason, which is a clump of about 30 to 60 cells, very rapidly after the initial fertilization event. And the guys in the audience in particular might have noticed that quantitatively that animated movie wasn't really very good. Because there's a sort of licentious chuckle from the depths here, because of course it just schematized 2 or 3 sperm, which of course is something like 100 million less than you would normally have when that event takes place, if all things are working well. So the reason for showing this is, first of all, to emphasize the beginning, as it were, from a single cell duplication to make multiple cells. But I might also, in passing, add that what happens to this clump of cells subsequently, very often at any rate, actually has some quite strong parallels with cancer, and in particular parallels with the way that tumour cells can dissociate themselves from the primary tumour, spread throughout the body, seed secondary sites and become what are known as metastases. And that's incredibly important because it's metastases, metastatic cancer, that accounts for about 90% of all deaths due to cancer. We'll come back to metastasis a bit later on. So both birth and metastasis are pretty fraught business. We don't actually know how many of these morula actually go on to produce babies in humans, but the fallout rate is very, very high. It may well be as high as 40%. However, with a following wind, and if you're lucky, a bit later it might turn into that, and even more scary than that, it might turn into this. And this looks, I know, it looks like a blatant plug for the book, and of course it is in a way, but I'd say it was taken at the Cancer Research Institute a couple of months ago at their open day by an oncologist colleague of mine who came past, and whilst taking the mickey, as you can imagine, also took from the pocket of his white surgeon's coat that piece of equipment without which no medic would be seen in public, and used his mobile phone to take this photograph. But the real point of showing this photograph to you is that what you're looking at, of course, is a clump of cells. It's quite a serious clump of cells because it's actually 50 million cells, more or less. That is to say, 5 times 10 to the power 13. And the point, of course, is that to get from our single cell a few moments ago being fertilized to a clump of that magnitude needs an awful lot of cell divisions. And the critical point in the current context is that every time a cell divides, two things must happen. First is that its genetic complement, its DNA, must duplicate itself, so you have two copies of all the DNA, two copies of all the chromosomes. And then the cell, having grown, effectively doubled in size, the cell itself and the DNA must be split in two to give two equal daughter cells. And I want just to spend a moment looking at that by showing you this movie. And we're just going to see a cell doing exactly what I've just described. And the cell in question is this one in the middle. You can't really quite see the outer boundaries of it, but you can see that there are 4 cells around it. And before I start the movie, you might note that this, of course, is the nucleus where the DNA is. The DNA is stained red in this movie. And you can see that the DNA here is a bit different to in these 4 cells around. It looks a little bit more squidged together and compacted. And that's because it's already duplicated and it's started to condense, is the term, ready for cell division. And the greens are the greens that we saw earlier. They're the proteins in the cell that do the heavy lifting, protein called tubulin, in fact. And we're going to just see tubulin in action. So here's the DNA. There are 2 poles that attach to the DNA, and the tubulin fibers, tubulin cables attach to the DNA and pull it apart, and then eventually the cell itself pinches off to form 2 separate daughter cells. And you can now see that the nuclei have reformed, or they're beginning to reform in here, and now they look much more like these because the DNA is now not condensed, it's gone into the state that it's normally in for most of the cell cycle. And one of the reasons for showing you that is to convey the notion that this is really actually a very physically violent process, and that, of course, was speeded up time-lapse cine. But nevertheless, it's a very mechanical process, as well as being an incredibly complicated one, self-evidently, if you think about the requirement of duplicating your DNA and then going through all the performance of pushing the cell through a cycle so that it duplicates itself. And you can look at that in a slightly different way. This is the same thing as a set of still photos, but here the green tubulin is actually coloured yellow, stained yellow. The DNA is blue, and you can again see the condensed DNA lining up on what's called the metaphase plate, being pulled apart by the tubulin cables. And these wonderful pictures were taken by George von Dassow in the University of Oregon. And even if you're not absolutely blown away by the biology, which you jolly well ought to be, what you can do is just have 2 cells instead of 4 and turn it into a greetings card. Okay. Now, as I warned you, I can't really manage to even get through that first phase of this talk without mentioning DNA and mutations. And I've pointed out that the cell division process itself, in addition to being mechanically really very violent, is also incredibly complex. And so there's a real potential for errors in every time a cell goes through this process of duplication. And of course, those errors, those mutations, potentially starters for cancer. But of course, in addition to that, a whole variety of external assaults can also have the potential to damage DNA, and we'll look at 1 or 2 of those a little bit later on. But having slightly put the cart before the horse, let's recap, and I suspect for most people in this audience it will be very much a recap, by just reminding ourselves of what DNA is. So DNA is what the chemists like to call a polymer. In other words, it's a whacking great big molecule made up of smaller molecules stuck together, represented by these coloured boxes here. There are rather a lot of these in our DNA genomic sequence, 3,000 million of them, in fact, in every human cell. And you will know that there are actually only 4 of these smaller units in the whole of our DNA. So it's a 4-letter code, essentially, that makes up our DNA sequence. And you can look at that, at least I hope you can, by putting in representative letters for the four-letter code, and these, of course, are A, C, T, and G. And so the smaller units that go to make up DNA, each of these boxes, themselves are made up of smaller molecules still stuck together, and the critical bit of those smaller molecules is the so-called base that gives rise to this designation of A, C, T, and G. But in fact, of course, as you will know, there are actually two strands of our DNA in all our nucleated cells, and those strands come together in the form of the celebrated double helix, held together by interactions between A and T and C and G. But I don't want to talk about the structure of DNA this evening. I really want to talk about the fact that DNA, or regions of DNA, can be transcribed into an intermediate, that's RNA of course, and thence read to produce a corresponding protein. And proteins too, as you will gather from my representation using Smarties here, are polymers, so they are made up of small subunits stuck together, but in this case the subunits are amino acids, and there are 20 different amino acids. So as opposed to a four-letter code for DNA, we have 20 individual units that can be put together to make proteins, but they can be used in any kind of sequence and they can be used repetitively. And what that means, the critical thing to grasp from this slide, is that you can make, in effect, an infinite number of proteins, and these can fold into an infinite number of shapes. And that's the reason for the variety of life on Earth. And if you don't believe that, or if you think that I'm using the word infinite in a slightly glib way, just do a simple calculation. Ask yourself, how many different proteins could you make if you just made the smallest, almost the smallest you could imagine? You made a little mini protein from 3 amino acids. You've got 20 to choose from. You can use them in any combination you like. Come back to that in a minute, maybe. But the critical point, of course, in the context of cancer is what's a mutation. So let's look at a mutation. Any change in DNA, any damage to DNA is a mutation, but we'll look at a specific one that changes just one base in this stretch of DNA, and then look at its effect on a protein. So, of course, what it might do is change the shape of a protein or change its function. And I showed here, I've gone rather rapidly through that set of slides, just the concept of thinking about proteins in the way that you might think about your hands. Just think about all the things that you can do with your hands and how flexible they are. And that set of photos was a rather celebrated sporting handshake. The cricketers will recognise Sachin Tendulkar as being one of the handshakers, and you saw him shaking hands at the Sydney Cricket Ground where it gets quite warm with that kind of hand, and you saw him trying to play cricket in England a bit later with his fist closed up and no suggestion of shaking hands at all. And I really like that kind of image of protein structure because it conveys the flexibility that proteins have. And in this set of slides, what I just tried to show was the effect of a single mutation caused in the amino acid sequence, caused by the base change that we saw on the earlier slide, and the way that that might give rise to a completely different structure in this protein, and hence to completely different function. So just to emphasize that point again, our protein structure may be changed by a mutation in DNA, even as small as one single base. And if that happens, the activity of the protein may be changed as well. It may become hyperactive, or it may be even knocked out. But an even more effective way of knocking out a protein is in fact to have a mutation that stops the protein being made altogether. There are lots of ways that you can generate that, and I'm not going to talk about those in any kind of detail, just simply to make the point that there are plenty of mutations that affect cancer because they essentially block the expression of a protein, and of course therefore you lose the activity. And if that sounds a little bit counterintuitive at first, the explanation is that there are really two factors that are at work in terms of, so let me just go back to that. There are really two factors that are at work in terms of regulating cell proliferation. There are factors that essentially are aiming to push the cell through the cell cycle. So they're, if you like, pro-proliferative drivers of proliferation. But there are also a set of things which are functioning as brakes, so that whether a cell goes through the cell cycle or not is a function of the balance between a set of drivers and a set of brakes, if you like. And so if you disturb this either by a mutation that makes a hyperactive protein, a hyperactive driver, turbocharging the acceleration, or you make a mutation that effectively knocks out the activity of the protein, either of these can be very powerful cancer drivers. And you could imagine that if you get two of these together in a single cell, you may have a very potent combination. Indeed, and indeed that happens quite frequently. Okay, so that's fine. And the upshot of all of that is that cancer seems to be really very straightforward. DNA damage may change a protein for better or for worse, so to speak, in terms of function. And the upshot of that is that it may tell a cell to grow when it shouldn't, the essence of cancer. But that, of course, then raises the question of, well, how do we talk to cells if these changes can tell a cell something or tell a cell not to do something? How do we talk to cells? And in principle, that's really quite difficult, because I suspect everybody will know that cells essentially are bags of fat, a fatty membrane, in the middle of which sits the nucleus where the DNA lives. And the membrane has evolved essentially to be impermeable to just about everything, apart from gases and actually, of course, steroid hormones. But we're going to keep off the steroids for tonight. And in fact, the vast majority of cells respond, or the vast majority of signals to which cells respond, are in fact extracellular messengers that normally, if one could imagine the cell looking just like this, wouldn't be able to get into the cell and talk to it at all. So how does information get across the cell membrane from these sorts of messengers? Well, the answer is that sitting in the membrane are a whole variety of receptors that act exactly as their name implies. They provide binding sites for the messengers, and they can then transmit information across the cell to an intracellular relay system of proteins. So it's very much like a relay team functioning to, first of all, recognize a change in the shape of the receptor as a result of messenger binding. And then when that happens, the relay is activated by the first part of the relay being able to interact with the changed shape of the internal bit in the cell of the receptor. And if that's a signaling pathway that stimulates proliferation, now you have a driver turned on by this growth factor, if you like, a pro-growth messenger interacting with its receptor at the cell surface. So this is information transfer rather than a physical transfer of an entity across the membrane, which is of course what happens with steroid hormones. So what about mutations? We've talked about how mutations might affect proteins in terms of their shape, but now suppose that we have a mutation in a receptor that signals to one of these relay teams that drives cell proliferation. And so I've shown that here by the green star, our mutated form, so that now, from the point of view of being inside the cell, all these messenger lads think that this receptor is being activated by its external messenger, except that it isn't. The mutation essentially has rendered the cell deaf to the outside world. It's a phenomenon that everybody in the audience who has children will recognize immediately. And so what that means is that the signal pathway is now turned on constitutively, as the jargon has it. And so you have, if this is a pro-proliferative pathway, something which is now out of control and which is certainly going to have the potential to be a cancer driver. So that's very straightforward as well. We've got a mutation in a receptor and that turns on a signaling pathway, and that's absolutely hunky-dory, and that shouldn't be too difficult to deal with. But if it is that simple, you might well be tempted to ask the question, why is cancer still killing 7 million people a year and the figure's set to more than double by 2030? Well, there are quite a lot of reasons for this. But one of the dominant ones is that we're actually living longer. So this just shows the increase in lifespan, average lifespan in the UK over the last century or so. And the critical point here is that if you look at the incidence of cancers, what this figure really tells you is that cancers are essentially diseases of old age. That is to say that 2/3 of all cancers do not manifest themselves in people until they've reached the age of 60. Of course, there are exceptions to that, and we all know about that. But for the most part, they are diseases of old age. And of course, what's going on in this figure is that because we're managing to live to these older ages, we're essentially making available more time to accumulate mutations. So one can look at it in the way that I do by considering life to be a game of genetic roulette. So one's parents do whatever they do and send you off into the world in a virgin state, and you trundle down the highway of life, and as you do so, your DNA acquires all sorts of bumps and dents and scratches and whatever. And that, of course, is absolutely fine. Rather like Homer's car, it really doesn't matter. Lose the odd wing mirror, smash the odd headlight. No odds at all. And the same is true with DNA mutations. Unless you happen to get unlucky, and by whatever age it is, you accumulate a critical hand of mutations that is a very powerful cancer driver. And then, and only then, do you have a problem. So that's one of the reasons why the cancer burden worldwide is increasing, because worldwide the population is living for longer, and that gives it more time to accumulate mutations. But there are other reasons. We do very silly things. But the reason I really want to talk about in the next few minutes is that cancer isn't quite as simple as we've made out so far. Okay. So we've seen that mutations drive cancers, and in the current jargon, the critical mutations have become known as drivers for very obvious reasons. They're the ones that are really important in terms of pushing cancers. And so tumours, we now know, need a set of these. Exactly what one means by a set isn't too clear, but the estimates range from somewhere like about 5 to maybe 15 driver mutations, real powerful cancer promoters. And those mutations are set against a very high background mutation in tumour cells. I'll come back to this in a moment, but typically tens of thousands of mutations in the cell. And clearly most of those are a little bit like the dents and scratches on Homer's car. They really don't matter very much. It's only if they arise in critical genes that they become important. So a model that I quite like is to think of cancer as a bus. But it's a slightly odd bus. It's trundling along a horizontal road, but it doesn't have an engine. So clearly, to get it started, and also, of course, to keep it going, somebody's got to be doing the pushing. That's the only way that it can possibly get moving. And in this schematic, clearly this rather attractive-looking young lady isn't the cause, because she's not going to be able to keep a bus going on her own, yet alone get it started. So we need something a little bit more beefy than that. We do indeed need sets of drivers, sets of mutations that really have a specific capacity to promote cancers. And I've represented that now with a sort of slightly more beefy push to our bus. And we've got half a dozen or so guys doing the pushing. And of course, each one of these represents a critical cancer driver mutation. And I've also shown, as you always get in these situations, a couple of people standing idly by with their hands in their pockets. And these are things that might become mutations if the fancy takes them. So the picture is really quite complex. And what it really means is that that if you think about this, the combinations of mutations that you require to drive a cancer, then you have even within cancers of the same type, and as we'll see in a moment, even within subdivisions of an individual cancer, you have different combinations of driver mutations. treatments. And that's really one of the major complexities in cancer. And what that means is that if you add that into the general background of high levels of mutation in cancer cells, that essentially every cancer is unique. And just think about that for the moment. Every cancer is unique at the biochemical level if you actually analyse mutations in the DNA. So what can we do about this? Well, historically, of course, as you know, the classic treatment for cancer has been surgery, usually with radiotherapy. And it's extraordinarily effective by and large. And the reason why you can say that is because about 90%, as I think I mentioned earlier, of deaths due to cancer are caused by metastasis, by tumour cells spreading to other sites in the body. And that tells you that surgery for primary tumours is actually remarkably effective. The problem then comes with our pushers and what we can do with them to deal with the problem of metastasis. And the only way that we can deal with that is by trying to treat the individual with drugs, the strategy known as chemotherapy. But the problem that we have is that until very recently, we really had only the haziest idea of what these mutations were. We really didn't know what they were. And so we had no idea how many of them you need to knock out. Do you need to knock out 1 or 2 or 3? And does it matter? Is it better to knock out this one and that one and that one? And even if you do that, what about the guys with their hands in their pockets? If you get rid of these pushers, maybe these guys become activated and start pushing in their stead. And indeed, exactly that, or the analogy of that, does indeed happen in cancers treated with drugs. So that's really our problem in terms of how we go about treating cancer. Particularly if cancers are identified at an early stage, surgery remains extraordinarily successful and effective. But if it is not identified early, the problem of dealing with metastases, with spread to secondary sites, at the moment really has no strategy for treatment. We have no strategy for treatment of that at all. All of which is pretty astonishing when you look at figures like this. Because against this background of really quite profound ignorance, over the last 40 years or so, there have been some spectacular improvements in the survival rates for some cancers. So in most of the developed world, the survival rate for breast cancer, for example, has gone from about 50% to now heading on towards 90%. For prostate cancer, it's gone from about 40% to over 80% in the UK. It's actually over 99% in the USA. And that's astonishing, really, because that's really been achieved by a combination of surgery and drugs on the basis of almost no knowledge of really what it is we're trying to do in terms of hitting molecular targets. It's a classic example of the way medicine has historically proceeded down the centuries, by trial and error. However, as you will know, there are other cancers that over this period of time have shown essentially no improvement at all in terms of 5-year survival rates. I've just shown lung cancer here, but there are many other, or a number of others of the major cancers that one can quote as well. And of course, what that tells you is that for all the ingenuity, for all the effort, and for all the expenditure that has been put into cancer research over that period of time, for these cancers, cancers like lung cancer, pancreatic cancer, for example, we've made really essentially no impact in terms of the clinical treatment of these diseases. My prediction, my fond hope is that when we look back at the beginning of the 21st century, we will actually see it as being representing a sea change effectively in the way that we look at cancer and the way that we treat cancer, and a complete change from everything that has gone before. And the reason for thinking that is that my greatest revolution is indeed the publication of the complete sequence of the human genome in 2003. And that happened quite coincidentally to be 50 years after Watson and Crick had come up with their celebrated structure of DNA. This is actually Francis Crick's wife's original sketch of the structure they came up with that was published in Nature. And the Human Genome Project really was an absolutely astonishing feat, an extraordinary triumph of international collaboration, admittedly predominantly between Britain and the USA, and also astonishingly because it contrived to come in under schedule and under budget, something that you don't very often hear in terms of anything to do with this country. But in fact, what has happened since the publication of the draft sequence of the human genome, the first human genome sequence, is, if anything, almost more astonishing. And on this slide, what I've shown is just simply the way that the cost of sequencing has come down, actually by 4 orders of magnitude, over the period of the sequencing project to date. But even more startling has been the technological improvements that have meant that the sequencing rate has gone up by something like a factor of 10 to the power 8. These are phenomenal technical advances, absolutely staggering in terms of what we can now do, even by comparison with 10 years ago in 2003. Now, I'm not going to talk about those or even show you them, but I have a very, I think, fantastic animated video that if people are interested, I can show you afterwards. But what I do want to do is to try and look at one example of the impact that the sequencing revolution has had on our treatment of cancer. So as I mentioned just now, enormous decreases in cost and increases in the rate at which we can sequence. And that, together with the development of astonishing devices for sequencing DNA by the means of fast throughput cells, we can actually now carry out hundreds of millions, and that is hundreds of millions, of sequencing reactions simultaneously at one time in one cell. And not surprisingly, this is a technique that's become known as massively parallel sequencing. Well, that's fine, but the interesting thing, of course, is what has it done for what we're talking about? And in a nutshell, what it now means is that we can take tumour tissue and we can sequence it repeatedly. In other words, we can sequence it in depth. And the point of doing that, of course, is like doing any other experiment over and over again. It means that you improve the sensitivity of the analysis that you're doing. And we can then have confidence in even the fine detail of the mutation pattern that we generate at the end of this repetitive sequencing. In other words, we can generate a complete mutational signature for a tumour, and we can do it now within 2 or 3 days at the cost of about $1,000. And again, that cost will come down. So you might suppose that one of the things that would come out of being able to carry out that sort of analysis is the identification of new cancer genes, and that may lead us to new drugs. Well, that's certainly true as far as the first part is concerned, and although we knew about quite a lot of mutations that cause cancer before the completion of the human genome sequence, we now know of an awful lot more. So new genes have emerged, and some new drugs have emerged on the back of that. And I want to show one example of that, which is kind of, if you like, the upside of the impact of the sequencing revolution on cancer. And I want to use malignant melanoma as this example. So it's not perhaps a cancer that may be all that familiar in this country. It's not even in the top 10 of UK cancers. It's the top of the tree in Australia, of course, because it's predominantly caused by exposure to sunlight. But nevertheless, we get quite a lot of cases in the UK, and perhaps most critically, it's the second most common cancer in terms of new cases in the UK for young people. So that tells you a little bit about their lifestyle, but it is a very important cancer in terms of its incidence. And you will know, I'm quite sure, that it arises from moles or birthmarks. We've all got them, and for the most part, they're considered to be either an adornment or at least not harmful. But if the appropriate mutations accumulate in those, then they can undergo the sort of change we saw on the slide. They can start to grow, they can erupt from the skin, they become itchy and they start bleeding. And if that happens to anything on your body, you really ought to get some expert advice about it. One of the very first projects that was carried out after the human genome was sequenced was indeed at the Sanger Centre, where the British component of the human genome was sequenced. And what was done was to take a panel of cancers and to apply the techniques of whole genome sequencing to them and to look for mutations. And in the group of melanomas that happened to be studied, something quite extraordinary came out of this fishing expedition. And it was a mutation that we now know arises in about 2/3 of all malignant melanomas. And it was astonishing because at the time we knew absolutely nothing about the genetics of melanoma. We had no idea of any of the genes involved. And the upshot of that, just go back and look at that again. The upshot of that analysis was to show that, that the critical gene that was mutated in malignant melanoma is a thing called BRAF. And BRAF is one of the internal messengers in the relay team that we saw just now. So these cells that have acquired a mutation in BRAF have indeed become deaf to the outside world. They need neither extracellular messengers nor receptor proteins on the cell surface. They have their own messenger system turned on constitutively in the cell. So that was an extraordinary finding, but even more extraordinary was that within 3 or 4 years it had prompted the development of a drug that is a specific inhibitor of the mutated form of BRAF, of the protein BRAF that drives this messenger sequence. And I just thought I'd show you a couple of PET scans here to show how effective this drug was in some of the first treatments of malignant melanoma, where the disease had already spread around the body. So this is a PET scan. Many of you will be familiar with those, and you can see that there are multiple dark spots all over this individual where the malignant tumour has spread throughout the body, and this is essentially an untreatable condition, except that this drug, the specific BRAF inhibitor, turned out to be remarkably effective at treating metastatic disease in this individual. It's the only example that I know of where metastatic disease has been really successfully treated, at least in a first-up treatment, with a drug. There are some problems with this drug in the longer term, I have to say, in the usual cancer fashion. But this was an extraordinary result, an extraordinary piece of fishing using the technology of whole genome sequencing, and then an extraordinary follow-up to produce an effective drug. So new cancer genes are one of the things that has come out of whole genome sequencing and our capacity to sequence tumours more or less at will. But the other thing that has emerged is really the full extent of the complexity of the genetics of cancer, what I call genetic mayhem. And I'm going to show you one piece of data that relates to this. It actually shows a primary tumour. It's actually shown as a kidney tumour here, but this sort of work has now been done on a number of other cancers. And the schema represents the notion that the primary tumour grows as they do, and that cells may spread from that to seed secondary sites, the metastases I talked about right at the beginning. And what was done in this study was that the primary tumour was excised, and then rather than taking the whole tumour mass and attempting to do genomic sequencing on that, what was done was to take very small samples, or rather like sort of taking little sort of cores out of the earth at different regions around the tumour, and to sequence each of those individually, and also to sequence one of the metastases. And the upshot of that was to come up with a cancer family tree for this single tumour. And this is an astonishing result if you think about it, because it looks very like the kind of species evolutionary tree that we're all used to looking at, where there are sort of chimps and apes and whatever, and we're in there somewhere. And in fact, it looks like that because that's exactly what it is. So what we're looking at is the development of mutations that drive this tumour effectively from the stage of the fertilized egg right back at the beginning over a long period of time where mutations are accumulating up to the point where we have what you might really call the most recent common ancestor. In other words, something that is genetically related to all the bits of the primary tumour that we looked at just now, as well as to the metastases. But the point about this figure is that the lines here are drawn to scale. So the length of the line represents the number of additional mutations accumulated from the point where it branches off. And in each of these cases, one's talking about tens of thousands of additional mutations. So this is a picture of astonishing complexity, even within one tumour. And it underpins the point that I made just now, that not merely is every tumour different, but actually even as we can now with modern technology probe within regions of a tumour, even those are genetically quite distinct from one another. Even though you can trace precursors and all of these guys, all of these regions of the primary clearly are related to the most common, the common recent ancestor. Nevertheless, they have diverged very substantially over the evolution of the tumour. So what does that really mean? What do we think is going on? Well, I made quite heavily, I think, the point before that the critical mutations that drive cancer development are so-called driver mutations. And they crop up in these signal pathways that we've looked at. And in principle, they can crop up anywhere in a signal pathway. So a mutation may occur in the messenger itself, so it's abnormal, and perhaps it binds to a receptor it wouldn't normally interact with or whatever. We've seen an example of a mutation in a receptor turning a cell on, and we've also seen an example in BRAF of a mutation in one of the relay proteins. So the point is that there are actually many of these signal transfer pathways, sets of information transferring molecules within cells. And that shouldn't be surprising because you will grasp, I think, very readily that cells are responding all the time to a wide range of signals. They're really rather like us as a whole organism. We respond to many sights and sounds and smells. And cells are really doing the same thing by responding to lots of different hormones and small molecules. And so in principle, if these different pathways all impact on proliferation, if they all are capable of giving a bit of a friendly shove to proliferation, you could argue that a mutation in any component of any of these pathways, signal, receptor, any of these bits, potentially is a cancer-promoting mutation. And indeed, that's the picture that we now have. So we can simplify that conceptually, I think. So forget about the individuals involved in here. And I find it now much, much easier to think about all of these things really as kind of like signal clouds. So there are zillions of signaling messengers, hormones and whatever it is. There are lots and lots of receptors, and there are lots and lots of these internal things whizzing up and down and sending messages to the nucleus, and so on. And in principle, mutations in any of these, or indeed in any combination of these, could drive cancers. What's the good news? Well, the good news is that because now we can do this incredibly profound sequencing of individual cancers, we can actually find out who's doing what. So we can actually find out which signals, which receptors, which relay proteins are mutated in a given cancer. And that's great, so long as we've got drugs that enable us to target those mutated proteins. And the problem in cancer is that we don't have those drugs. I showed you one, the inhibitor of BRAF, and I only know about 2 or 3 others that I would consider to be specific for mutated proteins. We have a lot of drugs that are not specific, which is why you get a lot of side effects with chemotherapy of cancer, but we have an almost bare cupboard of really specific targeters. So that's one of the big challenges facing cancer at the moment. And the other one that goes hand in hand with that is that even if you had a cocktail of anti-cancer drugs to treat a specific cancer and you applied them, it may well work, rather like our anti-BRAF drug did, for a period of time. But because of the inherent mutability of cancer cells, you can bet your life that the cell will find, or one of the cells will find a way around your drug, and it will use a different messenger, or it will mutate another receptor, or it will do something. And one way or another, you'll have an emergent clone, rather like the branches on our family tree, that's now resistant to your drug. So that's a huge problem for cancer. And the question that you might ask, therefore, is there another way that we can actually look at this problem? And I think if you think about this for a moment, you might think that actually there's perhaps a rather obvious way of looking at it. If these various combinations of signals and receptors and relays in the end are telling the cell to proliferate, or at least contributing to the instruction to proliferate, surely for something as fundamental as that to the cell, these multiple inputs must feed through a single kind of master regulator. And the answer is that it seems that they do. And the master regulator is a gene called MYC. And MYC's important because we know that you need the expression of MYC, you need MYC protein to get cells to go through the cell cycle, to be able to proliferate. So the point that we've reached here is that there's been a great deal of play about the notion of how DNA sequencing and the ability to dissect what's going on mutationally speaking in these clouds gives rise to what's being called personalized medicine. So we can take an individual tumour and we can say this is its signature and this is the therapeutic strategy that it requires therefore. The fact that the cupboard is a bit bare on therapeutic strategies slightly by the by. But the other way of looking at this problem is to say, well, maybe we should be a bit more radical. Maybe instead of thinking about personalised medicine, we should think about impersonal medicine. If we have a master regulator like MYC that is essential for proliferation and is anomalously activated, turned on, hyperactivated in probably all human cancers, but certainly most of them, how about targeting MYC? Well, the guy who asked the unanswerable or the unaskable question is a chap called Gerard Evan, and I call this last bit of this talk a glimpse of the future. Actually, it's also a serious look at the past, because Gerard's a very old friend of mine, and he's worked for many years in the University of California at San Francisco, and he's just come back to Cambridge as head of the Department of Biochemistry. And it was Gerard who, now many years ago, formulated this, what until then had been really considered to be the unaskable question of whether in fact we could systemically inhibit MYC and whether that might be an alternative anti-cancer strategy. And it's fair to say that when he floated that notion at one of the research group meetings, there was a pretty unanimous response from the assembled luminaries. And they thought that even by Gerard's standard, it was a pretty silly idea. And the reasons why it seems to be a silly idea is that, first of all, it's almost going to be bound to be toxic for any of the tissues that are being renewed at a high rate almost all the time in our bodies. So remember that as we, each of us here at this very moment, simply to maintain the status quo, we're actually making about 1 million new cells every second. That's 1 million every second just to stay as we are. So obviously, if you block cell proliferation by blocking the activity of a critical cell proliferating agent, the chances are your animal is going to drop dead fairly promptly. And in any case, cells that are really important in terms of keeping the system going, stem cells that make cells of the immune system and so on, you're going to do an enormous amount of damage to them. So it's just not going to work. It also won't kill tumour cells because if what you have is something that simply blocks the activity of MYC, they're going to sit there until that goes away, and then they're going to start growing again. So at best, it's going to be cytostatic. And there's another problem with MYC in that it's not like it's not like the BRAF example that we saw just now, because the problem with MYC is that when it's abnormally expressed in cancers, the mutation that gives rise to abnormal MYC actually just makes more of the normal protein. It's just overexpressed. The protein very rarely undergoes mutations itself, so it's not really a cancer target on its own. Nevertheless, Gerard persisted and came up with his reasons why we actually ought to go ahead and see if we can block MYC. And that's what they were. So somebody went ahead and did the experiment. And I'm just going to show a couple of molecular biology slides now. So for those of you who aren't into that, just look away now and look back for the results in a minute. But the point is that MYC is a master regulator. It's actually what's called a transcription factor because it controls the transcription of other genes. And like most transcription factors, it does it by interacting with a partner, whimsically called MAX. So these two proteins interact with one another, and then once they're stuck together, they can migrate to interact with the regulatory regions, usually called promoters of a gene, and turn on transcription of the gene next to the promoter. So that's how transcription factors work. How do you block MYC activity? Well, you make a molecule called OMO-MYC, and the idea for doing that came from a very gifted Italian scientist, Laura Susek, and the fact that she's Italian is not completely irrelevant to the story, because what Laura did was to make a molecule that was very similar to MAX. Actually, it was a homologue of this region of MYC, so that it bound to MYC rather like MAX did, but instead of allowing MYC then to activate transcription, what it does is in fact block the action of MYC. So now you have something that competes with the normal activator and blocks transcription. And Laura, being Italian and having built this homologue, called it OMO-MYC, pardon my Italian, and of course that was far too much for the English folk in the rest of her group, and so they immediately changed this into OMO-MYC, and as OMO-MYC it's passed into the literature. So that's how we can block the activity of MYC. But how can we do it in animals, in mice? And for that you need to make a transgenic mouse, because you need to be able to look in an animal at what goes on when tumours are generated. You can't do these experiments in the lab in a cell culture setup or something like that. And the strategy for doing that is now to put two artificial genes into a mouse. So it's a double transgenic mouse. One of them, of course, is something that makes OMO-MYC, and the other one makes an activator protein. And for that, you use a promoter that is going to be turned on all the time in the cells of the mouse. Doesn't matter what that is. But because it's turned on all the time, the cells make the activator protein all the time. And the activator protein, of course, binds to the promoter of this target, but it too only works when it interacts with another molecule. And the other molecule is a thing called doxycycline. Sorry about that, it's slightly ahead of myself. The other molecule is a thing called doxycycline. And the advantage of doxycycline is that you can put it in the drinking water of mice, and so you can essentially switch on the activity of the activator protein depending on whether you have dox in the water that the mice drink or not. And so here's doxycycline added to the water, the activator protein is turned on, and the downstream gene, OMO-MYC, is turned on, and MYC is blocked. So the first question is, now that you have a mouse system where you can reversibly inactivate MYC, what happens? Do the mice drop dead as everybody predicted? And the answer is no, they don't. And astonishingly, systemic inhibition of MYC is really remarkably well tolerated by the mice. There are a few side effects, but the side effects that there are actually turn out to be fully reversible. So one of them is that mice rather like us, if you give them a haircut, their hair will regrow over a period of a few days. But if you do this when the mice are on doxycycline, therefore they're making the MYC blocker, their hair doesn't regrow. So they have a bit of a problem in regrowing hair until you stop activating OMO-MYC and their hair growth starts again. They have some other problems. The males are infertile, for example, but they don't know that they're infertile. They're perfectly happy and they do what males do, seemingly completely unaware of the fact that they're infertile. And perhaps they shouldn't worry because, again, if you stop administering doxycycline and switch off OMO-MYC, their fertility returns within a few days. So OMO-MYC is fine. Now the question is, what effect does blocking MYC have on the development of tumours? And now we need a tumour model. So now we make a double transgenic, or it's actually a triple transgenic, because we cross our OMO-MYC mice with mice that can generate lung cancer. And this is a classic model for lung cancer in mice, and it's brought about by the activation of an oncogene called RAS, a cancer-promoting gene called RAS. It's a mutant form of a RAS gene. And you do that in much the same way as the trick with doxycycline that I talked about for activating OMO-MYC. So the strategy is to cross these two mice together and to produce a transgenic in which, first of all, you can turn on mutant RAS, and the effect of that is to generate tumours in the lung, and then you can turn on the inhibitor of MYC. So this shows the kind of results that you get with this experiment. And these are a little bit difficult perhaps to understand at quick glance, but these are sections of lung tissue. And if you look at normal lung tissue stained with something that shows up tumour cells bright purple, it really looks like a sort of fine piece of lace. And there's really not much stain on that tissue section at all. Now turn on mutant RAS, your cancer-promoting gene, and very rapidly, within a few weeks, these mice develop lots and lots of lung foci. And if you leave these mice alone, now expressing the cancer-promoting gene, what will happen is that these tumours will grow, they will eventually overtake the lung completely, and the mice will succumb to complete respiratory failure. What happens when you activate the anti-MYC, OMO-MYC? Well, the answer is that within 6 days, not merely do these tumours stop growing, but they disappear. It's an astonishing result. There's a little bit of purple staining around here, but that isn't tumour cells any longer. It's actually invading cells like macrophages that are mopping up the bits. So that's an extraordinary result, because it tells you that a single treatment with the anti-MYC strategy really makes a major impact on the survival of mice in this lung cancer model. And you can look at that data in the form of a typical plot that just shows mice expiring over a period of time. And here, when you turn on the cancer gene RAS, essentially all the mice are dead after about 30 weeks. Treat now with the MYC inhibitor, single treatment, OMO-MYC, and now about a third of your mice actually survive for at least double this period of time. Gerard likes to make the joke that when you show this data in Britain, the result is greeted with sort of dismay, and everybody sort of said, well, that's absolutely terrible experiment. 2/3 of your mice have died. Whereas if you show the same data in the States, everybody says, gee, that's fantastic. 30% of your mice are still alive after this treatment. But of course, there's an obvious experiment which will have occurred to you that one can carry out, and it's the last thing I really want to show you tonight. And it's quite astonishing, because you might say, well, if we've shown that one exposure to this anti-MYC agent, to OMO-MYC, can be tolerated by the mouse, that whatever the side effects are, they're not actually life-threatening or drastic, and they're reversible. What happens if we give it successive exposures? And the answer to that is that you can perfectly happily switch on OMO-MYC and block MYC again and again and again and again and again and again. And if you do that, what happens to the mouse death rate? And the answer is that the mice survive 100%. It's an astonishing result. Completely contrary to what everybody expected, that a central signalling element like MYC could actually turn out to be an excellent therapeutic target. It's also worth just commenting that, and we don't entirely understand this, but this extraordinary effect arises because we know now that normal cells and tumour cells have slightly different dependencies in their requirement for MYC. And that's why that you get rather little in the way of side effects from this drug, but very dramatic anti-cancer effects in terms of essentially the complete blockade or reversal of these lung tumour growths arising from the expression of the mutant form of RAS. And so, just two last points that this experimental model in mice shows, and that's that in thinking about cancer, it's so complicated, as we've seen, there are so many factors that are involved, that the only way that you can pin down therapeutic targets is actually to do the experiment. And you have to do the experiment in animals. You cannot do it any other way. You may do some of the preliminary experiments in cell culture, but the critical experiments you have to do in mice. And then finally, and maybe a little fantastically, let us make the assumption that mice and men are the same. That's a pretty drastic assumption in the cancer field because the highway of cancer therapy is strewn with the corpses of rat experiments that have been, and mouse experiments that have been incredibly successful seemingly, and yet the treatment simply hasn't worked in humans. But let's make the assumption. And if that is true, and that we can come up with an alternative to a genetic method of turning on an anti-MYC. In other words, we can make a drug that has the same effect as OMO-MYC, and we haven't done that yet. Then maybe we have the possibility that, for example, perhaps at age 20, we could offer everybody a dose of therapy, a short dose of therapy with anti-MYC pills. And then we say, go away and come back in 5 years, or maybe 10 years. And if we do that, and this works in the way it seems to be working in the mice, and bearing in mind the fact that abnormal MYC expression characterizes certainly the vast majority of human cancers, maybe we have a way, without any surgery or any radiotherapy or anything like that, of essentially preventing the initiation of essentially all the major cancers. And given that I've now strayed from the realms of hard science into the realms of fantasy, I think it's high time I thanked you all for being a wonderful audience and stopped. Thank you. I should say, ladies and gentlemen, I'm very happy to answer questions if people would like to ask them now by waving your hands or shouting out or whatever, fine. If there's anybody who would like instead or as well to chat individually afterwards, I'm very happy to do that for as long as it takes until they actually throw us out. And when they do that, we can always go around the corner to the anchor. But there was a gentleman at the back. Yeah, it's a very good question. It's not per se because cancer cells replicate more rapidly. It's an obvious thing to think about, because if you think about bacteria, very much in the news this week, one of the reasons why bacteria are very good at developing resistance to antibiotics is that they have very short doubling time. So you can force bacteria to double in 20 minutes in a petri dish. You can't do that with mammalian cells. But in any case, it's something of a myth that cancer cells proliferate rapidly. They don't actually, and quite a lot of them divide really very slowly. And what's more, if you look in a tumour mass, the kind of primary tumour that I showed earlier, all the estimates suggest that only a small percentage of the cells at any one time are really dividing at all. Most of them are actually pretty quiescent. So it isn't that. What it is that when you look at the family tree of the acquisition of mutations, it's in something that becomes a cancer precursor, there has accumulated a mutation or two or three or a set of mutations that now makes the cell a bit unstable. So by simply slightly deregulating the control of proliferation, cells now may go through the cell cycle when they wouldn't. And if you have mutations in your DNA already, then those tend to be amplified. So you get a phenomenon called genetic instability, which is exactly what it sounds. That really means that cancer cells are just more prone then, once they've started to accumulating mutations. And as you saw from the family tree, they do it in spades because these mutational numbers are phenomenal. It's not true of absolutely all cancers, but most solid tumours, you're talking about tens, even hundreds of thousands of mutations within the genome relative to adjacent normal tissue in one tumour. And the implication of that actually is that if you looked at individual cells, each individual cell in the tumour would be different too. So, Terry.
Can I ask, is the MYC gene very heavily conserved in humans, or do you find quite a lot of variation which could evolve itself?
Across species, you mean, or?
I was thinking within, for instance, you're treating a cancer, is that likely to have variation from cell to cell within it? And then could that actually be used, as it were, by the cells to produce a sort of colony?
Yes. Okay, so the question is, if you treat MYC, if you block MYC, how might a cell find a way round it, really? And the preliminary data I've talked about so far suggests that actually that doesn't seem to happen in the mice. Historically, with cancers, you would bet that that's what will happen. But it's a little difficult with MYC because there are very few, that MYC has two close relatives in the human genome, but they don't seem to have overlapping functions. So it's not easy for the cell to switch to using one of MYC's relatives, which is certainly what happens with, say, the receptors that I was talking about. So a lot of people in the audience have probably heard about things like the EGF receptor, particularly because it's a very commonly mutated gene in breast cancer. And if you take receptors like that and you have a drug that blocks their activity, what the cell will do, what a cancer cell will do, is simply switch pathways. And you'll find it's upregulated a relative of EGF, the EGF receptor, that does something pretty similar, essentially turned on another pathway. There's no evidence at the moment that that happens with MYC, and it's not easy to see how it would happen. But I bet something like that will happen. I mean, it always does with cancer, because what you're doing, I mean, talking about bacteria and the mutability of bacteria, the only real difference is that you're dealing with cells that don't divide as rapidly, but they're astonishing. I mean, you look at the flexibility in the human genome, it is astonishing. So I think that will happen. I mean, just on that point, it's worth making the comment that cancers at the molecular level are actually very rare diseases. Because when you think about the genetic mayhem that I referred to, usually what happens is that as mutations accumulate, something gets mutated that mucks up something that's fundamental to the cell. Can't make energy anymore, or its cell membranes get leaky, or whatever. And the cell just dies. So for a cancer cell to emerge in the teeth of all this genetic mayhem really is an astonishing singularity. It's kind of counterintuitive because we think of cancers as being so common. At the molecular level, they're actually very rare, or relatively rare.
Sorry, I'm going to be a bit cheeky now because we do need to move you out. But Robin is going to take his, well, however many it was, 50 million cells outside.
They're staggering to the door as you speak.